now we'll mainly focus on genome-wide, or the progress in genome-wide association studies and the new challenges that are ahead of us. So just to set the stage, we know that 40 to 70% of the variation you see in body mass index in this room is actually due to the fact that we differ genetically. So that means that there's a substantial genetic component that contributes to your genetic susceptibility. Yet, despite this substantial contribution, it's been very hard to find actual genes for obesity susceptibility. And in the mid-1990s, we started off with candidate gene studies. And candidate gene studies, as Julie yesterday showed, are hypothesis-driven. So you start from what the physiologists and the biologists know about obesity, based on their animal studies, extreme cases, or cellular systems, and then they inform the epidemiologists to tell them which genes they think are important in the biology of obesity, and say, well, these are maybe the genes that you should study for genetic variation in the overall population. And for many years, we've been trying to find candidate genes, and in the latest update, which is now already 5 years old, the latest update of the candidate gene approach, the human genetics map of obesity, more than 130 genes were proposed to be associated with obesity. However, only a handful have been really confirmed to be associated with obesity. And then later on in the mid-1990s or in the late 1990s, genome-wide studies were introduced, and that was because genotyping became cheaper and just more accessible to the larger scientific community. And genome-wide studies are hypothesis-generating, so it's the epidemiologist who screens the whole genome without any a priori hypothesis, and hope to find genes, or at least loci, that harbor genes, and then go with these genes to the physiologist and say, well, here's a gene that we think is linked or associated with obesity, but can you figure out what the biology behind it is? Yeah? So there, the flow of information is the other way around. It goes from the epidemiologist to the physiologist. And although Tony just showed you very convincing results of genome-wide linkage studies, I think the field in the last 15 years has been a bit overwhelmed by studies that have typically a small sample size and maybe not that well phenotyped outcome, such that convincing results have not always been replicated throughout across many studies. So overall, you can say that for the last 50 years that we've been searching for genes, it hasn't been that successful period. So, but everything's changed in 2005 when the genome-wide association studies became available. And genome-wide association studies are also hypothesis generating. So again, we screen the whole genome and hope to identify genes that contribute to obesity, and then go to the physiologist to find the biology. What's the difference with genome-wide linkage is that now we can screen the genome with a much greater resolution. In fact, we can study up to 2.5 million genetic variants across the whole genome. Another difference is that the study design is typically more robust than ever before for genome-wide linkage or genome-wide association, in the sense that whatever is discovered is always also replicated within the same study. So the end result is typically very convincing. And also the significance level that we aim to achieve is very stringent. In fact, we consider an association only significant when a p-value reaches 5 × 10⁻⁸ or less. And scientists have also realized that they needed to collaborate not only to get more out of their investment, but also because we realized that we needed big samples to identify the small effects that we can expect with genetics. And statistically, genome-wide association studies is fairly straightforward. And already in the mid-1990s, Neil Reich said that, well, genome-wide associations is the way to go. The question is, so why didn't we do it in the 1990s? Well, because we couldn't. We didn't have the technology. So the reason why genome-wide associations has come about in 2005 is very much technology-driven. Yeah? Particularly progress in genotyping technology that has driven genome-wide association study. Progress in genotyping technology allowed us to sequence the human genome and allowed us to identify the genetic variation across populations, and that's been cataloged in the human genome map and in the HapMap. And that information, which is publicly available, could then be used by companies such as Illumina and Affymetrix, who could make sensible chips to sequence the whole genome. And as Louis yesterday already showed, genome-wide association studies have been very successful. So the latest map here shown reported 904 loci that reached a significance level of 5 × 10 to the minus 8, 465 traits. So you see here the 22 autosomal chromosomes and also the X chromosome. And somebody asked yesterday whether the X chromosome is being studied. Well, apparently it has been studied, but not as consistently as before. And that's most likely because men have kind of inconveniently have only one X chromosome, so that sort of messes up the statistical analysis, or makes it at least slightly more complicated. But at least you see over the last 5 years an enormous progress in genetic epidemiology, enormous progress in genetic discoveries, and that's also the case for the genetics of obesity and obesity-related traits. And I'm going to take you through the different waves of studies in the obesity field. So you all may remember 2007, the very first genome-wide association study that reported that FTO is really robustly associated with obesity. And that was reported by Tim Frayling and colleagues in Science. And just a few weeks afterwards, Angela Scuteri also published a genome-wide association study. And we can consider these as 2 independent studies that both discovered FTO. And interestingly, the first study is actually a genome-wide association study for type 2 diabetes, whereas the second study is indeed the first genome-wide association study for body mass index. And then that same year, Anke Hene and colleagues, they performed a case control analysis with extreme early onset obese cases, and they confirmed basically the importance of FTO. And in the past few years, there have been many studies that indeed have confirmed the contribution of FTO, not only in white Europeans, but also in individuals of different ethnicity. And until to date, FTO is still the locus with the biggest effect size. It has not only the biggest effect size, but it's also very frequent. In fact, 64% of the people in this room carry at least one risk allele. And 16% of the people in this room carry 2 risk alleles. So it's pretty frequent. So the frequency combined with the effect size makes that FTO was fairly easy to identify. And here I show you the effect size of FTO. In fact, I've now expressed the effect of FTO in weight, in grams. And I'm expressing here the effect of 1 risk allele. So you can have a maximum of 2 risk alleles. And you see here that FTO, on average, carrying 1 risk allele of FTO, on average, increases your weight with about 1 kilogram compared to somebody who's got no risk allele. And that was in 2007, and we realized, at least in Cambridge, we realized that if we wanted to identify more than just FTO, we needed to collaborate with other groups to increase our power. And that's what we did. We combined our forces with people in Oxford and also in Exeter, and we performed a meta-analysis of 16,000 people. And that was indeed led to the discovery of the locus near MC4R that Jose just described. At the same time, our colleagues in London at Imperial College also did a genome-wide association study, this time in Indian Asians, and they also identified this near MC4R locus. We were at the time we discovered this MC4R locus, we were very excited because it was the second obesity susceptibility locus. But if you look back, you can wonder, why is it that we needed a sample size that was 4 times as large as the previous sample sizes to identify just 1 new locus? Well, the reason is likely because the MC4R locus is not that frequent, at least not as frequent as FTO, and the effect size is smaller. So that makes it harder, and you need more power to identify that locus. Under the knowledge that we needed big sample sizes, we decided that for the next round we've got to form a consortium, and that's indeed the GIANT consortium. And that's the GIANT consortium stands for the Genetic Investigative of Anthropometric Traits, and it's coordinated by Joel Hirschhorn at the Broad Institute in Boston. And that consortium had access to data of 32,000 individuals. And that led in 2009 to the publication of the Willer et al. Paper, where we reported the identification of 6 new loci very robustly associated with BMI. At the same time, our colleagues in Iceland performed a similar size study, and they identified 7 loci, although 4 of these 7 loci overlapped with the data that we reported in the Willer et al. Paper. And to give you an idea of the effect sizes, you see that because we had more power, we were able to identify variants with smaller effect sizes. Now, in 2009, we also started doing GWAS studies for waist circumference with the idea to find genes for more abdominal obesity. And that led to the discovery of 3 new loci. And our colleagues on the CHARGE consortium also performed a slightly smaller genome-wide association study for waist circumference, and they identified 1 new locus. And then also David Mayer and colleagues, they performed a genome-wide association case control study with, again, extreme cases compared to controls, and they identified 3 new loci. And just a few months ago, Andre Sherak and colleagues, they also did a candidate gene analysis, not a candidate gene, a case control analysis of extreme cases and controls, and they identified 2 new loci. Although these loci were not completely new because they overlapped with some previously identified loci. So I went quite quickly through that, so let me summarize this. 2007, we identified FTO. 2008, we identified the near MC4R locus. 2009, Willer et al. Identified, besides FTO and MC4R, 6 new loci. Thorleifson identified another 4 on top of the previous ones. And then the waist circumference paper, they confirmed FTO and MC4R, but they also found 3 new loci. And I put a little star on this one because that locus was associated more with waist-hip ratio than it was with waist. And I'll come back to that later. Then the colleagues from CHARGE, they identified, they confirmed MC4R and FTO, and they found a 3rd one. And then Andre and David Mayer, they did case control studies, and they identified a few new variables, a few new loci for obesity case control studies. And then just a few weeks ago, we reported on our probably our last genome-wide meta-analysis by the GIANT consortium. We call it the mother of all meta-analyses, in which we studied 125,000 individuals in a GWAS study, and we confirmed the result in another 125 individuals, and we identified 18 new loci for BMI. Just to give you an idea of the effect size, the effect sizes get smaller and smaller, but the effect size didn't reduce as much as we thought they would reduce. And in fact, you see quite a few loci that have effect sizes that are larger than the effect size that we identified in the previous round. And you can wonder, why is it that we didn't identify them at that stage? Well, that is because the frequencies of these loci is lower than the ones that we identified here. So these are the 32 loci for BMI. At the same time, within the GIANT consortium, we performed a genome-wide association study for waist-hip ratio, and waist-hip ratio adjusted for BMI. We aimed to identify genes for more the pure abdominal obesity, irrespective of the overall size. And that resulted in another 13 loci, and if I present them here in the effect sizes, these are the 13 loci. It's a slightly different scale as I showed the BMI loci, but in terms of effect size, it's similar to those of the BMI loci. But what's more interesting for the waist-hip ratio is that we see for 7 out of the 14 loci, actually, we see sex effects. We see that the effect for 7 of the loci is more pronounced in the women than it is in the men. Yeah. Very consistently, it's always the women that show a bigger effect size than the men. And maybe that reflects some sexual dimorphism that we see in actually the body composition of men and women. So to complete this cartoon, the 32 loci that we found in the last paper confirm the loci that were associated with BMI of the last rounds, but on top of that we found 18 new loci. And maybe I could point out, particularly to Tony, that this locus actually also harbors the POMC locus, quite consistent with maybe the linkage locus they found. And then the waist-hip ratio loci, the 13 loci, by definition almost, they do not overlap with the loci of BMI, but they did overlap with this one locus that we found before. So the most, what I think is interesting of this cartoon is that they all, from the start, found MC4 and MC4R and FTO. What's also interesting is that these loci, identified by case control analysis, were not replicated in any of the BMI or waist circumference loci. So maybe that suggests that for the more extreme phenotypes, different genes are at play. So in less than 4 years, we found 50 new loci really very robustly associated with obesity susceptibility traits. So what have we learned? And I'll give you first an overview of the last 2 papers, the paper on BMI and the paper on waist-hip ratio. And what we've learned from all these studies is that the variants that we identify have small effect sizes. The explained variance is pretty low. In both cases, the loci combined explain less than 2% of the variation in the trait. No wonder that the predicted value is low, and I'll come back to that at a later stage. Interesting is that the sex difference is only seen for the waist-hip ratio. We don't see that sex difference for the BMI loci. I didn't show you these results, but we see for the BMI loci are associated with insulin resistance and type 2 diabetes and HDL, but the waist-hip ratio are more often than the BMI loci associated with these traits. So maybe that supports the idea that people have that waist-hip ratio is more important as a metabolic risk measure than BMI. What we also have seen from few expression studies that we've done in silico expression studies is that the BMI loci seem to function more in the brain, regulate appetite and food intake, whereas the waist-hip ratio, the first results suggest that they may have a role more peripherally in adipose tissue metabolism or adipocyte metabolism. So these are first insights. I'm going to show you a few more results, not of the 32 BMI loci, not of the 14 new discovered waist-hip ratio, because these results have just come out and we haven't had time to do any further analysis on these, but I'll show you results of the 12 loci that we identified last year. And first question that we asked, so these loci were identified with GWAS studies for adults. So the question is, do these loci affect childhood or maybe birth weight? So this is a result that has just been accepted, where we did a meta-analysis of the results of these 12 loci for their association with birth weight. So we had data on almost around 24 to 28,000 individuals. And again, you see the effect size for each allele, and now the y-axis is in grams. See, these are the 12 loci, and the error bars are the standard error bars. And you see our conclusion of our paper was that there's actually very little or no evidence for association of any of these loci with birth weight. If you want, you can say, well, maybe FTO is associated, the risk allele that increases your body weight also increases your birth weight, but the p-value is not extremely convincing given the sample size that we have. In some cases, you see even the opposite, which people who believe in the Barker hypothesis of in the fetal programming might get excited about. Although overall, we think that these body mass index loci do not affect birth weight, at least not to a very relevant extent. So then the next question is, well, do they affect childhood obesity? And again, we did a meta-analysis, and this time it was a meta-analysis by Marcel. These are the results of the adult effect sizes, the ones I showed you before, but I've now standardized them so that I can compare them with the childhood data. So the y-axis is the standardized effect size for increasing BMI. And these are the result of a meta-analysis of about 14,000 children and adolescents of these 3 studies. And you see that overall, many of the adult BMI loci already have an effect in childhood. For example, TMM18 seems to have even a bigger effect in childhood, as SEC16B as well, although some loci have not yet an effect. MTCH2, SH2B1, they seem to have only effect later on in life. Now, these are cross-sectional studies. I showed you one data set on birth weight, one data set in children, and another data set in adults. Ideally, you have longitudinal data for that. And that's what we did that's an analysis that we did for FTO and MC4R. So for this study, we had data of 2,500 individuals born in a week in 1946. They were all born in the same week in 1946. And across the years, they have been called back for further measurements during childhood, adolescence, and adulthood. And we examined the effects of the FTO variant on BMI at different time points. And you see here the age at the x-axis, and at the y-axis you see the standardized effect of the 3 genotypes. So the 3 lines show you the 3 genotypes. And as you see, early on in life, there is no effect of FTO on body mass index, but soon after birth, you could say, the effect of FTO starts to increase, and the body mass index of the 3 genotypes basically diverge, and they reach a maximum difference at the age of 20, and then the difference sort of reduces again. And we saw the same effect for MC4R. So the question here is this a typical pattern, the way body mass index loci work? Or is this just a coincidence that FTO and MC4R show the same pattern? What does it mean? Why is it that the effect is larger at the age of 20? Is it maybe because individuals stop growing and after that the environment sort of takes over the control of body mass index? Or is it a generational effect? Individuals born in 1946, they will be 20 in 1966. 70s, sort of the increase of obesogenic environment. So these are very interesting results, but it also raises a lot of new questions that we would like to follow up using other birth cohorts across the years. This has been shown before, where we genotyped these 12 variants in 20,000 individuals of the EPIC Norfolk study. This is the same study that was discussed before. And we made a genetic score. So if you have 12 variants, if you're really unlucky, if you have 12 BMI variants, if you're really unlucky, you inherit 24 risk alleles of your parents. If you're really lucky, you inherit none of them. Yeah, so it's a range between 0 and 24. And most of the people carry between 10 and 13 loci. And 1%, 2% of the population carries 17 or more, and 1 or 2% of the population carries 6 or less. And if you then plot the BMI according to this risk score, you see very nice increase. So each additional risk allele increases your weight by about 440 grams. And interestingly, we've done the same analysis on the 32 loci, and in a different study, and we see that the effect size is nearly the same. Yeah? So despite increasing your genetic score, there's sort of no leveling off. It seems that the effect is consistent. We have to keep in mind, of course, that these extremes are pretty rare. And as Louis and Claude yesterday showed, when we split the population in active and inactive, then you see that in inactive individuals the effect is larger than in inactive individuals. People ask us as well, can we use these 12 variants to predict whether a newborn baby will be obese in later life or not? So what's the predictive value of these 12 variants? To express the predictive value, we typically express the sensitivity of a test against the 1 minus specificity of a test. So how well can your test identify obese individuals, and how well can your test identify non-obese individuals? So ideally, a perfect test has a perfect sensitivity, perfect specificity, so the area under the curve is 1. If you get a lousy test, as was said before, if your area under the curve is 50, then it means that's as good as flipping a coin. So how predictive are these 12 variants to predict obesity? Well, this is the result. Just slightly better than flipping a coin. Yeah? So we cannot use these 12 variants to predict whether a newborn individual will be obese later in life. Again, interestingly, adding these another 20 variants of the latest paper, it does not improve this area under the curve. The area under the curve is actually exactly the same as before. So these are nice epidemiological studies, give you an idea of sort of the public impact, or the impact on public health. But I think these genome-wide association studies are at least equally important to pinpoint towards new biology, or to point towards new biology. And the question is, so what have we learned so far of new biology? Well, not extremely lot. So FTO has been around the longest. FTO has been around for 3.5 years, and that's the gene or the locus we know the most about. It's highly expressed throughout. It's highly expressed in many tissues, but it's predominantly expressed in the hypothalamus. The expression is influenced by fasting and feeding. It's associated with food intake in both mice and in animals. And a study a year ago showed that if you knock out FTO, then the mice are small and at the same time have an increased energy expenditure. So there is evidence at this point that it works centrally as well as peripherally. Yeah? So, but I think there's a lot more research that needs to be done to really figure out what FTO means. And of course, not only what FTO means, but what many of these other new loci mean in terms of biology. A bit more information. We know that some of the newly identified loci and some of the previously identified loci harbor genes that were previously associated with monogenic forms of obesity. So that's where the monogenic and the common genetics sort of converge. As I pointed out, POMC is harbored by one of the loci, MC4R you knew already, and also SH2B1. Copy number variants that have some people have been pessimistic about copy number variants, but we found 2 loci that perfectly tag copy number variants. We also identify a locus that, quite interestingly, the BMI increasing allele is associated with an improved glucose, insulin, and metabolism, so maybe the fat but fit gene. So that provides you already the first insights in potential biology behind that gene. We've done some bioinformatics, and it was not extremely successful, so that hasn't yielded any convincing pathways. We've done some gene expression studies, and again, the results some are interesting, but it hasn't yielded really, or it hasn't helped us pinpoint towards the causal gene. So what's the challenge ahead? Well, we hope to find more genetic variants, and we do that by refining the phenotype. Tony just pointed out that you can increase your power by using more accurate phenotypes closer to the biology, and that's exactly what we're doing. We've just finished the genome-wide association study for fat percentage. We're doing a genome-wide association study for intermediate traits as well, such as food intake, and you know that's very hard to measure. as well as physical activity. We are doing genome-wide association studies in children, as well as the Asian community has formed a consortium, and they are now going full force to find genes in Asians. Because of genetic differences, they might find different genes, or maybe the same genes but with different effect sizes. We are also doing genome-wide association studies for interaction, so do interaction studies at the genome-wide level. But if you really want to be sort of ahead of science, then you need to start looking for rare variants. And that's what many people are doing right now. The belief is that, okay, we found a lot of common variants, but maybe the rare variants are even more important. And if you don't have a lot of money, but you want to participate in finding rare variants, then you can fairly simply participate by using the data that's available from the 1,000 Genomes Project. And the 1,000 Genomes Project is just sort of an extension or a more refined map compared to the HapMap, who report 15 million genetic variants and also 20,000 copy number variants, and you can impute that data into your GWAS data. And that way you can potentially find new genetic variants as well. But if you have a lot of money, then maybe you should invest in sequencing. You can sequence the exons, but you can also sequence the whole genome. And the current belief is that this type of studies, looking for the more rare variants, might point towards new genes, maybe genes or genetic variants with bigger effects. But that's still sort of an open question, and the next few months and the next few years will tell you whether that's indeed true. And while I think finding new genes is indeed a worthwhile effort to do, I think we should be careful that we not forget about the loci that we have discovered, because remember, we've discovered all these loci, so we've got 50 loci, but we want physiologists to follow them up, right? And I think a major challenge remains in that sense that it sounds simple. We have a locus, and we call the locus MTCH4, for example. And you see here, this is a close-up of the Manhattan plot of MTCH4. You see on the y-axis the p-value, and on the x-axis you see the different SNPs according to their position, and this is chromosome 11. The top hit is located right here, so it has a p-value of 10 to the minus, in the meta-analysis, 10 to the minus 9. And the top hit is located right on top of MTCH4, and that's why we call this locus the MTCH4 locus. But if you look right here, all these SNPs here, these dots here in red, they're in perfect LD, they're in perfect correlation with our top hit. This basically means that any of these genes located under these loci could be the causal gene, right? So there are about 14 other genes that could be the causal gene. So if I go to physiologists and say, well, the top hit is on top of MTCH4, but it could be any of these 14 genes, it's not very helpful. So the main challenge is even for these established loci, we still need to find out, we still need to define what's the causal gene. And once we know what the causal gene is, then physiologists can follow it up and do some more experimental research towards the biology. So I think that's one of the major challenges that we may not forget to follow up on, apart from only finding new variants. So I'd like to summarize here and say that over the last 4 years we've made enormous progress, found 50 new variants associated with obesity and related traits. Explained variance is small, and predictive value is low. What I think is very important is that hopefully some of these newly identified loci will lead to new biology, even if only 10% or say 20% of these loci lead to new biology. I think it's already a success. And as I said, for many loci, the challenge remains to really find the causal variant. And I'll finish here, and I should say I'm extremely grateful to be part of, oops, I need to show you all my collaborators. It doesn't work. But these are all my collaborators, about 400 people. So I feel extremely grateful to be part of the giant consortium, and particularly to these 15 people who've been really the driving force behind these latest papers. Particularly Joel, who is the coordinator of the giant consortium, but also Liz, Kristen, Caroline, and Karen, and also Mark. But I have also a great group in Cambridge with people like Marcel, who's done the childhood data, Thomas, who's done the birth weight data, Shanks, who's done a lot of the genome-wide association data, and have very important statistical support from Jenan and Jinhua. And I'd like to finish here, and I think it's time for questions.
Okay, thank you.